All right. So thanks again, everyone, for being here. We're going to start with a general short introduction to organic IPM for soil-borne diseases. So really foundational question here. I just want to start with what is a pest? Generally speaking, it's an unwanted organism that damages or interferes with plants. So usually we think about insect pests like cucumber beetles, but pests really encapsulate all kinds of different organisms like rodents, weeds, and pathogens, which is the subject for today. So what is IPM? You might have heard this before. IPM stands for Integrated Pest Management. So it's integrated because it combines multiple strategies instead of just like relying on one thing. And the goal really is managing your pest populations and keeping the damage under your thresholds. And I say your thresholds because that might look different depending on who you are and your context, goals, and constraints. So just bear in mind that can look a little different depending on your farm. And IPM is really this big picture, holistic ecosystems-based approach. So there's this emphasis on long-term pest prevention to start with, which I think is really powerful. And this IPM is really a toolbox approach where you have all of these different tools and strategies, and you're going to choose the ones that really make sense for your unique context. So there's no one size fits all solution here, but you do have a lot of options in your toolbox. Also, within IPM, in this approach, you really only use pesticides after monitoring indicates they're needed. And of course, you always want to follow the label directions. And if you're going to go this route with pesticides, you'd use strategies to minimize risks in non-target organisms as well. And so traditionally, IPM is kind of presented in this hierarchical triangle diagram where you start with, at the bottom, identification and understanding the pest that you've got, and then moving into different types of control. So maybe cultural, sanitation, physical, mechanical, biological, and then chemical is at the top, really highlighting like, okay, we want to try lots of other things first, combining techniques and really reserving chemistries only if they're really needed. You can also think about IPM though as kind of more of a cycle where you're kind of always doing a lot of these things, and it's maybe a bit more cyclical. So there's just different ways to think about it. But the idea is that over time, you're going to refine your approach. So I wanted to point out the UC IPM website, which is really great. You can start with your crop type. For instance, let's say cole crops, and then use online resources to help identify your pest and assess which strategies actually really make sense in your context. As you can see for like cole crops, we've got a whole list of diseases that are found in California. They've got a list for pests, insect pests, and other things too. So that's a nice resource. But please do bear in mind, we need more California-specific research on soil-borne disease IPM, specifically in organic systems. So as you know, I'm sure you guys have probably seen this website before. Maybe you've noticed this, but that's why we have Gabriel here, and we'll get more into that later. So within this IPM framework, we're going to start with inspection and identification to correctly identify the pest, make sure you know what you've got, and scout and keep good records over time, even if it's just like a notebook or a sketch or something simple and easy. And then you're going to look at different resources online, reach out to me, and then choose your actions accordingly. And then really assess, did it work? Did it keep the pest under your defined threshold? Maybe it's a certain number of bugs per plant or bugs per sticky trap or something like that. And then over time, you're going to reevaluate and refine your approach based on what you see and observe and evaluate. So I'm going to go through just some quick examples of soil-borne diseases of visual signs and symptoms. So this is just to get us thinking about like, what does this stuff really look like in the field? So we're going to start with damping off pathogens. So this is a complex of multiple pathogens like Pythium, Rhizoctonia, Fusarium, Phytophthora. So it could be any combination of these guys. And what happens here, we have these stems that are attacked near the soil line, causing young plants to collapse. So the nice thing about this one is that it's pretty characteristic. So if you see the base of stems looking kind of pinched, like you do with these red arrows and all of these different plants, that can be a really helpful characteristic sign of damping off. So that's kind of like a first look at it. Switching over to Fusarium in tomatoes, Fusarium shows up in a lot of different types of ways in tomato plants. You might have Fusarium wilts, which is, yellowy, wilting leaves. It might be Fusarium crown or root rot. A nice way to look for this is just slice open the base of a plant. You do have to sacrifice one plant, but if you slice it open and you've got this disease, you're going to see dark vascular discoloration, like where these arrows are. So that's pretty characteristic. And then you could also have Fusarium foot rot, which basically looks like, shows up like this sort of dark discolored root tissue. And so this is all Fusarium, but it's different types of Fusarium. And so Gabriel's going to get more into that later. But this is just kind of a first look at like, what does this stuff look like in the field? So Fusarium and strawberries. We have these pictures here, really nice symptoms. Well, it's not a good thing, but very helpful symptoms here of sort of like cinnamon colored discoloration if you slice open the crown of a strawberry plant and you see this. And also above ground symptoms just look like leaf die off. It sort of starts with, a part of a leaf or a couple leaves, and then it can progress to entire plants. However, with Fusarium, it's a lot less characteristic. So a lot of different types of diseases in strawberries can cause this kind of wilt. So we'll get more into sort of distinguishing things in a moment, but there's so many more examples out there, but that's just kind of a first look. So let's say you see some symptoms of Fusarium in strawberries, or you're just like not totally sure, you want to know with a lot of confidence what you're dealing with, you can look into pathogen diagnostic labs. There's quite a few out there. You just want to consider cost. You can always just call ahead of time or look at their websites. There's a really nice selected plant and soil labs in Northern and Central California. This is on UCNR website. It's a downloadable table of like tons of different labs if you want to compare them. Just bear in mind that we're talking about diagnostics labs for pathogens, which is different than like a lab that would analyze like soil fertility or something. So a separate thing. But I know like growers have mentioned they use like ALNL labs or TriCal diagnostics. So those are just examples, definitely not necessarily endorsements, but I know growers use those a lot. Okay, so what are like soil-borne diseases tend to have in common? There are some common themes. Broadly speaking, they often thrive in certain environmental conditions, such as moisture, humidity, rain, flooding, poor soil drainage, compaction, clay soil. So big theme here is water, of course. This diagram nicely shows the more soil compaction you have, the less pore spaces, the more water to air ratio. So pathogens would love the situation on the right here more than the one on the left. So, this is broad, and in some cases there are differences in preferences between pathogens. So especially when it comes to temperature, some pathogens, thrive at different ranges, temperature ranges. But this water theme is pretty common. Another big one is plant stress. So plant pathogens are gonna love stressed plants because they're just easier to colonize. That might mean maybe the plant's already nutrient deficient or water stress, maybe the root growth is restricted, and maybe it's already damaged by other pests. Like this tomato plant here had whiteflies, spider mites, aphids, and also Fusarium. So there's a lot going on. And on the flip side, plant pathogens can make plants more susceptible to other pests too. So just bear in mind, a lot of times there's more than one thing going on. And lastly, a lot of these different soil-borne diseases produce a ton of different spore structures that can survive in the soil and on plant tissues, sometimes for years. And so there's this really diverse reproduction strategies. On the left here, we have diagrams of different types of spores. And then on the right, this is what some spores look like in real life. So these diseases produce a lot of inoculum, unfortunately. So briefly, we're just going to go over some general IPM principles for soil-borne diseases, starting with prevention. So genetics. If you want to get high quality, vigorous seeds and transplants that grow quickly past that susceptibility phase when they're really young, you can also explore tolerant or resistant crop types and varieties. And strawberries are a good example where we do have some pretty successful resistant cultivars, whereas other cultivars are a little bit more susceptible to specific diseases like Fusarium. There are limitations there too, though, if you have a market for a specific variety. I know that can be the case sometimes, but it's an option to explore for some crops. Location's another big one. Ideally, you want to plant somewhere where there's no history of soil-borne diseases. Of course, that's not always possible. But if it is, I think that's a really good strategy. Also, crop rotation, diversification can help as well. Not planting the same thing in the same spot every single year, if you can help it. Sanitation is big, so regularly disinfecting stuff, whether it's tools, greenhouse benches, work areas, anything that you reuse in the greenhouse or start house, like pots. Sanitation is big. Growing conditions, so germinating your seeds at a relatively high temperature, so seedlings grow pretty quickly out of their most susceptible phase, using potting soil that drains really well, pasteurized, and especially if you have diseases in your field, please don't use field soil, for transplants and starts. And of course, just avoiding overwatering is helpful. So anything you can do to improve good drainage and soil aeration will help to some degree. So, I know some folks on pretty small scales, and if that's the case, I think a broad fork can be helpful. It is labor intensive for sure, and I'd only recommend it on a tiny scale, but it does help with aeration. On a larger, mid to large scale, you can diversify root architecture through cover cropping or crop rotation, and just generally minimizing activities that compact the soil, like traffic, equipment, stuff like that will be helpful. And good plant health. Anything you can do to ensure sufficient nutrients and water will be helpful, because in general, stress plants are going to be more susceptible plants. Another big one is promptly eliminating sources of inoculum. So removing and discarding diseased plants immediately is really going to help because they are a source of inoculum or spores that can affect nearby plants. So that, as soon as you start to see symptoms of disease, be just, if you can, pretty quickly removing that out of the field is going to be helpful. Soil solarization is an interesting one. So this means, during fallow periods in the hottest part of the summer, putting clear plastic tarp on moist, bare soil for 4 to 6 weeks. So this can help reduce pathogens to some degree. And in general, many beneficial soil microbes can survive or bounce back pretty quickly. So this is an interesting one. I think it's worth trying out, especially just starting in a small area first and seeing how it goes, I think could be helpful. So some biological options, cover crops. So there are some studies that suggest they can promote the suppression of some diseases by influencing the soil microbe community, increasing beneficial ones. But we do really need more research to understand like how this works, and then that would influence, how do we use this most effectively to control diseases. So that's a really exciting area. So we do have some organic fungicide options that, some products are based on hydrogen peroxide. We have some organic biofungicide options, using different microbes here like Streptomyces, Bacillus, Trichoderma. So these are all products that use microbes to help fight these soil-borne pathogens. And of course, adding compost can always help promote beneficial soil microbes, like after you use something like this too. But again, always consider cost, is it really worth it? Some of these products I think are pretty expensive. So that's just something to pencil out for yourself, for your farm. And once again, we need more research on these types of products and their efficacy and how to use them most effectively. So just to summarize your soil-borne diseases IPM toolbox, with an emphasis on organic, starting with visual ID, maybe send to a diagnostics lab if it makes sense for you. Starting with good genetics, thinking about location, rotation, sanitation, growing conditions, drainage, good plant health, getting that inoculum out of the field as quickly as you can. Maybe trying out soil solarization on a small corner, see how it goes. And maybe trying out some things, if it's within bandwidth, such as biofungicides and cover crops. Again, we really need more research on it, but I know a lot of you are already trying this stuff out, so I'm excited to hear about how things are going. So just to summarize, IPM is a process, things come up, you get curveballs all the time with farming, so just bear in mind, it's a process. And just to reiterate, we need more research, so we have a lot of major knowledge gaps. And we're focusing on Fusarium today because it was the most common one last year in our region. A lot of growers I talked to found that they have it. So that brings us to Gabriel's presentation. And just know that I'm rooting for you and you can reach out anytime. I think you all have my email. I can also give you my phone number. And with that, we'll go to Gabriel's presentation. If I can figure out how to use Zoom. Oh, here we go. Stop share.
Thanks for introducing me, Ellie.
Yes.
As Ellie mentioned, I will be giving a presentation diving deeper into the specific management tactics we can use for Fusarium. First things first, I want you to know that it's not just you, and it really is difficult to control Fusarium, and there's a lot of interest in that where I am as well. And I am trying to figure out how to reverse this. There we go. So again, my name is Gabriel Sacker. I'm a postdoctoral researcher at the USDA ARS in Salinas, where I work for Peter Henry. And I am also kind of in the labs of Nicholas LeBlanc, who is also in Salinas, and Sharifa Crandall at PSU. So there's a little bit of funding for me from a lot of different spots, but it also means I have a lot of expertise to draw from. As I mentioned, we're located in Salinas, which is definitely part of big ag. That comes with a lot of benefits and a lot of problems as well. And it means that a lot of our research and focus are on larger scale production types. But I'm going to try to make sure everything that I mentioned today is applicable on a smaller scale and definitely for organics. Historically, Salinas has been pretty cool. So we have a lot of strawberry, leafy green, and brassica production. But as the climate is warming, it's changing what we can grow here, and is also changing the diseases which we face, which includes Fusarium wilt, which is becoming more and more of a problem. So again, our lab focuses on strawberry pathogens. I'm pretty much appointed just for Fusarium wilt. That's how big of a problem it is. And we're exploring some of the epidemiology, so how it's spreading around. Maybe there's some aerial dispersal components to this. And we look a lot at genetic resistance. Having difficulty advancing the slides. So Fusarium oxysporum is the causal agent of Fusarium wilt, which is sort of the classic and most common Fusarium disease we see in diversified vegetables. But unlike many pathogens, Fusarium oxysporum is mostly not pathogenic. And so it's honestly the weird strains which are infecting our crops. It's just that they're so common and we spread them around as humans that we find them in a lot of our fields. So each of these Fusarium oxysporums kind of has to be dealt with by itself. And so there's many different strains, and they often only infect a single crop or maybe very closely related crops. For example, we'll have like the lettuce pathogen, it only affects lettuce. Or maybe we'll have the Fusarium oxysporum, which infects cabbages, but maybe that one will also infect broccoli because they're so closely related. Sorry, guys. And because of this, we need to really focus on good diagnostics. On the right side, I have two diseases here, and one of them is verticillium and the other one's Fusarium. And it's basically impossible to tell them apart in the field. So I really encourage you, if you think you're suffering from Fusarium wilt, to use a diagnostic lab. Because again, the visual symptoms can look so similar, but then the IPM that we need can be very different. I really encourage you as well, when you get those diagnostic reports, to focus in on the maximum specificity. If they just tell you it's a Fusarium, right? Well, I just told you that most of the Fusarium oxysporum isn't even a pathogen. So we really want to see like, okay, what's the genus, the Fusarium? What's the species, oxysporum? And then if we can get more detail, like what is the specific strain that we're facing here, that can help us determine what host crops it's going to affect. And it's possible you'll have more than one in a field soil. And then again, I'm keep hitting on this because it can't be overlooked for Fusarium, that the pathogen is diverse, which means that our outcomes when we're trying to fight this disease can be diverse too. And something that works really well against one strain might not work against another strain. But I'm going to try to introduce you to the tactics that are most well supported, and so you can try it in your own field and see what's working in your context. All right, now I'm going to start the more management section of the talk. And I'm really going to be hitting here on disrupting the disease triangle. So for any plant disease to occur, it has to have a susceptible host, there has to be a pathogen present, and then the environment needs to be favorable to that pathogen. And so by interrupting any of these parts of the disease triangle, we can make the disease less bad. And I'm going to start here with the host. As Ellie mentioned, we have resistance. In strawberry in particular, it's really good, but you can find this across many different crops, and it can be a silver bullet. For example, in strawberry, you can see on the top left, if you plant a resistant cultivar, it looks great, even in the same soil that's absolutely melting other types. And so many of these crops will have complete resistance. In cases where there's not complete resistance, there's usually a spectrum of susceptibility where some crops are affected to a moderate degree or not at all in some years, where other cultivars, can you see my mouse? Perfect. Where other cultivars are basically melted regardless of the year and regardless of the pathogen pressure. So you can find these resistant cultivars, hopefully at your seed supplier. You can look for these codes. Sometimes they'll call like VFN, verticillium, fusarium, and nematode resistant, or some subsection of that. They often won't get more in depth about like what specific strains or races are this variety has been tested against. But the more detail that they give you, the more confident you can be in sort of the vetting that the cultivar has been through. All right, now we're going to talk about the pathogen side of disrupting our disease triangle. So the more fungus there is in your soil, well, the more of the pathogenic fungus there is, the more disease you're going to get. And that kind of makes sense intuitively. If there's just more of it there to attack your plant, it makes sense that it would damage your plant more. And so we can use management tactics to reduce the amount of pathogen there is in the soil. And this is sort of an example of how much of a difference you can make. Even with pathogen present at a certain low level, you can see no disease. But then across different cultivars of lettuce, you can see as we get 10 times more pathogen, 10 times more pathogen, 10 times more pathogen again, you see more and more plant mortality. So that will really vary anywhere from 0% to 100% of your plants being killed just by the amount of pathogen which is in the soil. So we can start to reduce that amount of pathogen through disinfestation of your soil. In a large scale conventional field, they're probably going to use fumigation, but obviously that's not an option for an organic farm. So we can use things like solarization, where we basically kill it with heat, or we can use something called anaerobic soil disinfestation, which is almost a biofumigation. So rather than using heat, we basically prepare a bed really well and spread a lot of organic matter, often something like rice bran, and we tarp it just like soil solarization, and we basically let the anaerobic decomposition that occurs naturally in the soil build up toxic gases, and that almost smothers pathogens. This works well for Fusarium if you're able to leave it for a long time, but it can also backfire and make Fusarium wilt worse. So I wanted to mention this because it works really well on shoulder seasons when it's not as hot. That might make solar sterilization not work as well. But if you want to try this on your own farm, I'd really recommend working with somebody who's done anaerobic soil disinfestation before to make sure it kills your Fusarium rather than just provides a lot of material for the Fusarium to eat. Now, a last organic method for disinfesting your soil would be steaming. It's relatively expensive, but the bonus is it's so quick. Solarization or anaerobic soil disinfestation take weeks, but steaming can take hours. It is mostly used in greenhouses, but you really can apply it anywhere that you have enough steam generation to heat. It offers great short-term benefits, but because it's such a hot kill of everything in the soil, sometimes it will clear the soil of everything, and then the Fusarium will come back first. And so you might only see short-term benefits. We can also use crop rotation to fight Fusarium and limit the amount that's in the soil. This is a graph of Fusarium populations in the soil with different crop rotations. So one thing that was really interesting is if you plant raspberry, you can get a huge increase in your strawberry specific Fusarium. So this is very crop specific. And then if you plant your strawberries, you tend to have relatively unchanging amounts of Fusarium. But then basically any other plant that they tested, all were contributing to reducing the amount of this strawberry specific Fusarium in the soils, with the best treatment being fallow. And so here's sort of a cheat sheet of everything they tested, where most of the crops barely helped Fusarium at all, and it died over time. Certain crops could be reservoirs for the pathogen, but not ever express symptoms. And then our susceptible strawberry was highly symptomatic and basically melted. So it gives us a lot of confidence that we can plant basically anything that isn't infected by the disease and get disease reduction over time. But we do have to pay attention for Fusarium strain specifics. So if we are planting strawberries, we have to check. It's like, okay, what does the research say are other plants we have to avoid? And then if we were doing, tomatoes, we have to check, what are acceptable rotation crops. And the answer is most of them, but it's good to double check. And then here's an example with lettuce. And so again, most of the crops they tested were okay versus lettuce. But interestingly, spinach had a high rate of Fusarium colonization, which was the opposite of the strawberry list, where spinach didn't show this effect. So again, it's a very crop and Fusarium dependent. The rule of thumb is 1 year out of your susceptible host is a good level of control, but 3 years is much better. And this only works if you get it before there's an outbreak. So this won't really remediate your soil unless you're really ready to spend a lot of time, but it will help sort of nip a Fusarium wilt in the bud if you get it early. Now we're going to talk about a favorable environment, the last piece of the disease triangle. First, Fusarium is a heat-loving pathogen. Generally, the warmer it is, the worse your Fusarium is going to be. But again, it can interact with other factors. For example, cultivar. We can see like here, a susceptible lettuce dies really quickly, almost regardless of the temperature, whereas a mildly susceptible lettuce sort of does okay unless it's a very warm temperature, and then a resistant cultivar will just never get the disease because it's resistant. And we can use this to our advantage. There's a lot of talk in early season about what we can do to raise or lower the temperatures of our soil. And if we implement some of those cooling strategies, especially as the summer warms up, such as mulching with, say, straw, we can reduce the temperature of our soil and suppress the disease a little bit. Of course, there's other consequences of having cooler soils, but this is in the context of Fusarium wilt. These are a couple maps where they mulched with a dark green plastic, which is really kind of rejects the heat, versus a clear plastic, which causes that greenhouse effect and really warms the soil. And everywhere that's red is basically a dead plant. So highly lethal under very warm soil conditions and, disease, but not as bad under cooler conditions. And I mentioned temperature because it's sort of the number one that's really drives Fusarium, but there's many other factors which have been tested. Many of them need more study, but here's sort of a little cheat sheet where an increase of pH, so a more basic soil, more organic matter, more nitrate as compared to ammonium, and more calcium in your soils will make the disease less bad, whereas higher temperatures, higher redox potentials, and higher levels of free iron and manganese will really make Fusarium worse. And this redox potential basically is compacted, waterlogged soils. So just like Ellie mentioned, we definitely want to avoid that. And none of this is a panacea, but by piecing these strategies together, we can start to reduce the severity of our Fusarium wilt. As you saw on that last slide, many of those factors are kind of fertilizer based. And I thought this was a good illustration of just the difference it could make. So this A is ammonium, this N is nitrate, and the I means the crop was inoculated, and this is cucumber. And so under high levels of ammonium, the plant is fine, unless it's exposed to the pathogen, in which case it's devastated. With the reverse, with high levels of nitrate, the plant seemed to be protected with the inoculated plant being almost as healthy as the non-inoculated plant. As organic growers, you don't really have access to nitrate fertilizers, and so this doesn't help you as much. But the important thing to note here is that a lot of the water-soluble organic fertilizers do have free ammonium. So if you're relying on that as a primary nitrogen fertilizer technique, you could accidentally be creating this situation here with high levels of ammonium and favoring Fusarium wilt. So how can we reduce the levels of free ammonium we're applying and really kind of have lower, longer release treatments instead of those quick flushes of ammonium? There's so much more to say on fertilizer, including raising soil pH, and we can talk about that more later. But the nitrogen dynamics are really the number one thing you should be looking at, as well as pH for your first two things, fertilizer-wise, to combat this pathogen. Organic amendment, obviously compost is great, but not all composts are created equal. I'm sure we've all had situations which we've ordered a way too much compost and it just wasn't good quality and we don't know what to do with it. And one of the things which kind of determines how good that compost is how old it is. But there is a sweet spot. If we get really fresh compost, it's almost, still just the component parts. And that has a lot of free material that the Fusarium can actually eat. Remember, this is a soil inhabiting fungus that doesn't necessarily need a host to survive. So if you're applying a lot of kind of fresh material to your beds, the Fusarium can eat that and make your disease worse. So what we want to apply is kind of this middle area here, where it's like a really well digested compost. And that is still active enough with enough kind of like nutrients and carbon in it that it hosts a robust microbial community that will suppress the Fusarium and not allow it to get hold, but is not too old where we end up on an extreme where basically we have peat moss that's so decomposed that there aren't really any microbes eating it anymore. And so there's nothing left to suppress the Fusarium. It won't necessarily make the Fusarium worse, but we're losing our kind of microbial ability to suppress the pathogen. One good example of a nice amendment is vermicompost. It's been processed by the worm and sort of increased in microbial diversity. So it has a good mix of like high in nutrients, but also highly colonized by things that aren't Fusarium. So the Fusarium can't eat it very well. And there's been several studies like where you inoculate an eggplant, for example, with Fusarium, and in a kind of more of a dead soil without earthworms, it's stunted. But with earthworms, we're getting some good fertilization effects and good microbial colonization, which helps the plant. And at the same time, we can apply just the worm castings where we have lower disease index, which is in the front, and higher plant weights, which is in the back, when we inoculate pots with 15 or 30% vermicompost, which is a lot, but it does illustrate that this can work. Finally, other amendments, brassica seed products, so like mustard meal and chitin have been shown to reduce Fusarium wilts. Neither of these are super effective, but they do see some reductions in disease pressure in at least small scale tests. There is some interest in using these in different kind of decomposition processes like anaerobic soil disinfestation to increase their efficacy. But one thing you should know if you use either of these products is you need to apply it at least 3 weeks before planting. For chitin, if you don't do that, if you just apply it right before you plant, it just won't work. But with the like mustard seed meal, it actually breaks down and releases a toxic gas in the soil. And so if you don't apply it early enough, you'll hurt your plant roots. So it's something to keep in mind. But both of these are just regular fertilizers, and so could be substituted for what you're already using. Now, some final thoughts. As Ellie mentioned, plant stress is really important. A lot of what I was talking about, especially in the environmental section, is just good crop management. We want good fertilizer regimes. We want good soil aeration. So it's doubly important to focus on those if you have Fusarium wilt. We also have plenty of biocontrols, fungicides, and defense inducers on the market, but none of these have really been shown to work, at least at an economically effective level against Fusarium wilt. They work better against some other pathogens, don't get me wrong, but not so well against Fusarium wilt. And so it brings me to one of my favorite webcomics. It's when you see a claim that a common drug or vitamin kills cancer, it sells in a petri dish. Keep in mind, so does a handgun. And I feel like that applies so well to agriculture too. It's like we have a lot of these, heavy hitters, but that doesn't necessarily mean it's going to work once we apply it in our farm fields. And it doesn't mean we'd want to apply it. And lastly, I want to encourage you all to do some on-farm research. So many of these things aren't sufficiently answered, and usually UC advisors or USDA personnel are willing to help you design some trials. I really recommend that you keep it simple, but make sure it's replicated so we have at least a couple plots and have a good control. And if you are suffering from Fusarium wilt, I'm definitely interested in talking to you about it more, or running some on-farm trials, something something like that. And that's my email there at the bottom. And that's all I have.
Awesome. Thank you so much, Gabriel. I think let's just go straight into questions. If that sounds good to everyone, we can, ask Gabriel some questions, and also feel free to share like anecdotes, what you've tried, what works, what doesn't. So yeah, feel free to kick it off, whoever wants to.
Yeah, and I want to second what Ellie said. A lot of the people I work with are much larger farmers, and they can just like slap a fumigant on there and call it a day. And so a lot of these things I'd really be interested in hearing what experiences people have on a personal basis.
I was wondering if you have seen any issues with like the soil solarization with like plastic, with that also killing any like good things in the soil that contribute to the overall like soil health or like root health of plants later? Like, does it set you back at all?
It's often the opposite. So by killing a lot of the pathogens, we're leaving room for the other soil colonizers that will break down a lot of what's already there. So we'll often see a boost in plant health after soil solarization. A lot of that is attributed to kind of greater turnover in your organic matter in the soil. And so we're sort of like it accelerating a lot of the organic matter breakdown. And that's why we want the organic matter there in the first place. So we're kind of draining the bank account a little bit of maybe long-term soil health, but it doesn't set our plants back. And that change in the microbial environment of the soil, not always, but sometimes will be more of a long-term suppressive effect on diseases, where it's just there's so many other microbes that we've sort of selected for that some of the pathogens can't get in anymore. And that's not to say that will be a permanent solution, but often we'll see it last longer than like a steam treatment, for example.
That was a great question. I'm taking notes in the share doc that I just put a link to in the chat, and anyone should be able to like put notes into. So if I get something wrong, just correct it. But yeah, awesome question and answer. Other thoughts, questions?
I can share a little about what I mean, Ellie had, I think I had a lot of, well, Ellie had told me how there's, different crops have the different species, but I didn't like fully, yeah, it's just interesting to hear that talked about in more detail. But I had gotten strawberry plants tested last year, but it seemed like there was, I mean, I had a wide variety of plants growing that showed similar symptoms. I have a very small space. I had been farming on a larger space, and now I'm I lost the land that I was farming in 2022, and my plan in 2023 was to focus on the most profitable crops in my own backyard space. So it's quite a small space, and I was mostly planning to focus on a couple of strawberry varieties and a bunch of cut flower varieties. And there's not a lot of, it doesn't seem like there's any any flowers being bred for resistance. And I don't know, but many of the flower varieties I grew showed symptoms that also looked like Fusarium rot. And then I had some random vegetables as well. And I feel like just at least 50% of what I grew of diversity of crops showed symptoms that looked very similar, but it was only the strawberries that were tested. And the previous year, I had grown everything, almost everything had grown very well, although there was a few, I think there was a little bit of, the strawberries didn't grow well the year before, and there was something that looked like some kind of wilt disease on some cucumber plants. But I had flooding, my backyard definitely had quite a bit of flooding. I also made my own compost, which probably wasn't fully broken down, so maybe that contributed as well. So I definitely have learned some things from your presentation, but I'm trying to figure out now. Well, all that I've done so far was I sort of stopped planting new crops, and then I had planted vetch and crimson clover as a cover crop, which I had just like Googling found information that said that seemed like they might be helpful in establishing a more beneficial fungal environment. So I guess now I'm just kind of trying to figure out like what makes most sense going forward. And it's quite a small area, but I would like to be able to use it as a production space. And I would like to focus on very high value, on just a few high value crops. So I don't know. Yeah, I'm kind of just like processing everything. I guess like one thing I do want to get like more details about how to like properly solarize, because that seems like an effective strategy. And also just like how to get the best, then bring in like the best compost that I can, that like is gonna be inoculating the soil with more beneficial yes, I don't know if I have like real specific questions, but that's sort of like the situation that I'm in. It would be nice to hear a little more detail about the solarization, or how you'd recommend like recommendations for that. You mentioned the shoulder season, but like exactly, but then it also, I think the slide said something about 86 degrees. That doesn't sound like the shoulder season where I am. So maybe more guidance on how you would recommend solarizing.
Yeah, I can say a couple of things, and then maybe Ellie will be able to jump in too. First off, I'm really sorry to hear that you lost your land. That's a huge bummer. And I also want to say that you're not alone with getting Fusarium on high value crops. That's often what gets people down here, is because the farmland's so expensive. You know, they'll grow like just strawberries or just basil or something like that. Mm-hmm. And then they have no way to rotate out to kind of get a different crop going. Mm-hmm. Something like solarization could be a great option for a small space where you can sort of just hit everything and sort of get a little bit of a reset going. For solarization, it tends to be sort of in the middle of the season. The hotter the outside temperatures are, the faster this soil is going to heat up and get to levels which are killing pathogens. For soil solarization, it's really important to prepare your soil as well as you can. So you're going to want to get kind of like a level seed bed, just like you were going to plant into it, and then water it really well, because that water is going to help transmit the heat better than air will. And then you cover it with basically a greenhouse plastic if you have it, or growers down here will get stuff that's much thinner because they will just throw it away after they're done. But any sort of clear plastic will work. And that's going to let the light in, but then trap the heat, and it creates like a mini greenhouse. And you're looking for temperatures of at least 110 degrees Fahrenheit. Usually it's 110 to 125. And you will take that out up to like 8 weeks. And I don't have in my head exactly like the target number of like heat hours to go for Fusarium. But generally, the longer you do it, the more of the pathogen it will kill and the deeper it will go. So I can definitely get like more specific sort of hour requirements. And a lot of that's posted on like UC and our websites as well. And then you want to seal the edges of your plastic as well as you can to sort of trap the warm air in.
Yeah, and I'll just add real quick, and then maybe we could go to Vince, but yeah, I think that was a great explanation. I don't think there's a whole lot I'd add, but I did just put a link in the chat to soil solarization. I will say there, maybe earlier there are other methods. Like I remember, Gabriel, you mentioned, adding biofumigants. So maybe that was where the temperature question might have been coming up, Rachel, where.
Oh, yeah.
Yeah, so like maybe there's like other temperatures or timing related to that practice. I think that might have been the shoulder season one. Was that right?
I think it was the anaerobic soil. Yeah.
So just those things being separate. I mean, you could even do both if you really needed to or wanted to. So yeah, let's go. Oh, go ahead. Sorry.
One more thing. You can combine them. So you can do soil anaerobic disinfestation under really high temperatures, and that's shown to work well too, but that's pretty new. So promising results, but I don't know how much I could point to you, like specific studies
Mhm.
that would address your context. Vince, I think you had your hand up first, if you want to go ahead.
Yeah, thanks. And sorry to join late today. So I hope my question wasn't already covered earlier, but um, I'm curious about transferring Fusarium from infected areas. Um, you know, if you're rotating crops and trying to prevent hosts from, creating a reservoir in an infected area, that's great. But if we're using the same equipment to work multiple blocks, or if there's like winter rains that carry water through infected areas and on into other blocks, are either of those vectors for Fusarium? Do we have to think about disinfecting equipment if we're trying to prevent it from spreading to other parts of the farm?
Yeah, that's a great question, and it is something of a lot of concern. One of the ways that Fusarium gets around the most is on people's equipment or infected transplants. And so doing your best to limit the spread by, say, pressure washing your BCS or something like that is going to help. One thing we do have going for us is if you are moving the infested dirt to other places without a host crop, it doesn't survive very well. And so you're not necessarily going to accidentally make a bed like unworkable to strawberry the next year. But at the same time, if you have a bed of strawberry that is infected and you work it, and then you move that same equipment into your other strawberries, that's definitely a way a pathogen can get started. One thing to keep in mind is the numbers that we're talking about for how much inoculum we need to kill a plant aren't really going to be moved around by you. It's the thing that really gets you is once the disease starts and it's killing the strawberry, it grows so much disease on that dying plant that that's what's going to last for a long time and cause you trouble down the line. If you're using the same equipment and you have a disease bed, it's probably safe to assume that you have at least some level anywhere you go. And so once you've tested positive for like a Fusarium of strawberry, you should be thinking about, okay, how am I going to limit these outbreaks just as my baseline cultural practices by extending my crop rotations, by finding cultivars that are less susceptible. So you never run into the problem where you've set up the perfect storm and just get Fusarium to run through your field. It also can travel in the water and potentially even in the air. So, if you're downhill from a infected field or something like that, definitely watch out. There's only so much you can do, and I get that. But the biggest thing to watch is transferring from an infected field to a susceptible field, because you're setting yourself up to, let the disease go.
That's great. Thank
Susanna? Oh, yeah, you're welcome.
Yeah, I'm wondering, like specifically for like something like tomatoes, if we use the same like infrastructure, like posts for trellising year to year, is that like something that even if it's like a year apart and it's stored, is that something that's like a risk for transferring? And then also, I know in one of your charts you were showing different crops that kind of were more desirable hosts for this. And so are there certain crops that you recommend kind of if you're able to kind of plan a rotation before and after a susceptible crop, like brassicas or something specific that you would recommend kind of trying to slot in before susceptible crops or after to kind of treat the soil? And then, sorry, a few questions. I know another part of your presentation was talking about how soil temperature creates an environment where this thrives. And for crops like strawberries or tomatoes, where we often are mulching the beds with plastic, I know straw is the alternative, but for labor reasons, often plastic is more convenient. And if that's kind of an unavoidable setup, is there something we can do to treat the soil before, or any tips?
Yeah, absolutely. So I'll try to take those in order. So first, with the pathogen remaining on some of your tools, it definitely will.
Okay.
That is something to worry about. But it's most important early in your kind of growth cycle. For example, if you're growing your own tomato starts, you really don't want Fusarium like on your greenhouse structure or on your trays. So focus on the sanitation more earlier in your crop cycle. So there have been studies, for example, to show that it will hang out in your greenhouse. And so one place for a fungicide is sanitizing your greenhouse structure. And there's organically acceptable like the hydrogen peroxide, xylitol, for example. And I don't have all the sanitizers in my head at the moment, but that might be something to check with your organic certifier about. But start early and keep it as clean as you can. And getting those healthy transplants in the ground will help mitigate the effects of Fusarium in the future, or at least delay it long enough where you can get an economical harvest before Fusarium gets you. If you were just talking about like in-ground, like trellising stakes, the soil is going to hold much more pathogen than like a stake will. And so it's probably not contributing to the pathogen or to the disease cycle by itself. But if you do something like soil solarization and you're like, not sterilizing, but you're cleaning the top 6 inches of your soil or so, and then you take a stake loaded with Fusarium and plunk it right in the middle of that, then you've just kind of re-inoculated your clean area. So if you're going to work on any of these kind of like cleaning tactics, then you'd want to make sure you're, cleaning your stakes that go into that. Yeah. And then I can't remember your next 2 questions already. I got too into cleaning things.
No, I appreciate it. Mulch, and like creating a environment that is warm and cozy for Fusarium. And is there any way to like treat the soil or anything to do after your beds are made and your plants are in the ground to kind of like make that less appealing?
Yeah, absolutely. So if you remember those maps I showed you, that was with plastic mulch. And so they used a clear plastic mulch that really warmed the soil, and that was terrible for Fusarium. Like it killed lots of their plants. Versus a dark green plastic mulch, which was able to keep the soil cooler. And I think they use dark green because it's not clear, so it's not causing a greenhouse, and it's not black, so it's not absorbing as much of the heat. And there's nothing particularly special about dark green, and you could look at like mulch suppliers and they might have something more to recommend. But something like that could be a tactic to still get your mulching effects at an economical price, but then not heat your soil. A lot of them will heat your soil at least a little bit though, so it is something to keep in mind. Yeah. And then what can you do after the plants are in the ground? The biggest thing I would say is so many of the tactics are best done before you plant the plant. So there's only so much you can do with in the ground. But some of the bigger things you can do is basically like what not to do. So it's like, don't apply high levels of free ammonium fertilizer. That'll exacerbate Fusarium. Don't let your plants get waterlogged or drought stressed, because those will open up to more Fusarium disease. Definitely do things like roguing, which is the removal of infected plants that are showing wilts. That might not necessarily save you a lot during that season, but it will spread up to say 4 tomato plants, I think is the best estimate, root by root contact. And so you can kind of slow that down, as well as remove some of that infected material for the next year, so it can help you down the line. That's more labor intensive, and so I think often people don't do it, and there haven't been a lot of studies on that, but leaving that disease reservoir where it can kill its neighboring plants is not always the best.
Right. Thank you. And then just sorry, the last question was about the crop rotation, if there's certain things that are good to kind of put before or after susceptible crops.
Yeah, so again, that's sort of like an avoiding thing where unfortunately we don't have enough like research to give like an ideal crop rotation strategy, especially considering how many different Fusariums there are. So it's going to be different for a lot of different crops. In both lettuce and strawberry, broccoli has worked really well, and I think you could expand that out to other brassicas. They have kind of root exudates, which are known to be toxic to a lot of different fungi. And so we often want to give a little bit of time between, we want to plant that crop, harvest it, or it's a cover crop, terminate it, leave it for a few weeks, and then come in after that to get the maximum benefit from the brassica crop. But because of all those, basically toxic substances in the roots, they also don't host Fusarium very well. And so it can sort of act as a good break. But given that there are Fusarium which affect brassicas, so this is sort of a good general advice, but if you get Fusarium in your brassicas, it's going to kill your brassicas. Again, it's a totally different strain, so it won't go back and forth, but it just kind of gives an example of how wild this can be. Grasses are often pretty safe. So like a wheat or rye shouldn't get infected very well by these sort of, vegetable pathogens. Again, disclaimer that different Fusariums will play differently, but they're often kind of different enough from the host that you're planting that they won't host like the strawberry Fusarium very well. And so that's a good option for like a high biomass, good for the soil cover crop that won't host Fusarium very well. Last thing, if you do grow a lot of biomass, sometimes that will feed the Fusarium. So just keep that in mind. If you are applying lots of undecomposed organic matter to a field of brassicas, that could be less than ideal.
Thank you so much.
Mm-hmm. Rachel, do you have a question?
Well, I was wondering if like wood chip mulch in pathways, if that contributed to things in it, if that could kind of be a problem or a benefit, and also just if there's any way to inoculate beneficial fungi, that like if that's a strategy that, I found like a little bit of information about that, but if that's something you're familiar, or like something you can like dip plants in before transplanting, seems very time consuming, but whether you had any information about that kind of thing, or something that you could mix into your compost.
Absolutely. Great question. So the wood chips to start, I think that is in general, is going to keep soil temperatures down. So I think that would help you. If you're leaving them just on the surface, like honestly, with soil particles, there's so much surface area in your soil that when we talk about the contact between soil and wood chips, they're not affecting the soil underneath so much, right? Mm-hmm. It's often through like, say, a worm that comes up and pulls it down that will sort of start mixing it in. But the Fusarium in the soil is not going to be able to eat a wood chip sitting on the surface so well. I definitely would not like mix them into the soil, because then you're making them much more available to the soil organisms. And then also like nitrogen tie-up, there's other issues there. But as just a surface mulch to reduce temperatures, that could definitely help. It also might help, just in terms of spreading on your equipment. I almost think of it like they'll use those gravel driveways at construction sites to knock dirt off. And so that can help clean boots and such to at least some degree. And it stops like soil erosion and stuff from spreading it. In terms of inoculating soils, there haven't really been any commercial products that have been demonstrated to fight Fusarium wilt in particular. I know that some people swear by like mycorrhizal inoculations and stuff just for general plant health, but I haven't seen anything that like you apply this to your plant and it reduces Fusarium wilt. One flip side of that is following, and this is sort of a limitation of science, is it's hard sometimes to get at little differences. And so we're looking at sometimes the most extreme examples. But if you fumigate your soil and basically, kill most of what's in there, you have a very not diverse soil that's susceptible for Fusarium recolonization. But if after you fumigate it, you apply vermicompost, and that's what this study did in particular, it changes the microbiome of the soil to be very similar to vermicompost and not similar to the fumigated soil. And that has resisted other pathogens. No one's done it on Fusarium yet, that I'm aware of, but it is something to keep in mind if you are doing any sort of like disinfestation treatments. Applying a really good compost like vermicompost can help re-inoculate that. And one of the reasons I think it's different is just the sheer amount that you're applying. Like a root dip can be beneficial, but you're not really adding that much to your soil. But if you're applying vermicompost, you could be, you're applying literally tons of microbes to the soils.
Thank you. Cool.
Absolutely.
I don't know how to do the hand.
I don't either. Don't worry.
But a couple questions. So you had mentioned mustard meal. So can we? Okay, so whatever we had in our field was affecting multiple different crops from different families: peppers, tomatoes, brassicas, lettuces, legumes, kind of like the whole spectrum of things. We avoided planting brassica cover crops because we had a whole crop of cauliflower and broccoli that was killed. But thinking about mustard as like a biofumigant, I mean, can that potentially be used as it does seem like the wild mustard seems to grow unaffected and healthy, which I don't know how to make sense of that, because it'll grow right in the middle of a bunch of brassicas that are being killed off. But we were considering doing a mustard cover crop and then incorporating that to see if it had any sort of like biofumigant qualities to it. So I don't know if that's a possibility, but on that same note, with the cover crops, and I talked to Ellie about this, but someone else I had talked to had suggested leaving a field bare for the winter, like not to cover crop it, to not give that food source to the pathogen. We kind of freaked out at the last minute, and we were just like, we don't want to leave it bare and have it just erode during the winter, so we did cover crop it. But now that we have these fields cover cropped, it's like, what would be, we do have the potential to move the whole operation to a different area, but as we like address the problem in the area that we have been working on, like what's our do you have any idea of like our process going forward? We have this cover crop, like, do we kill it and just let it kind of hang out on the surface? Do we try to incorporate it and then do the solarization? And then in addition to that, I guess the last question would be tilling. It's like we did primarily for the most part like no till on our farm. And we do have a rototiller that I'm not opposed to using it at all, but we just didn't do it. But is that a bad thing to till it, or does that tilling potentially bring, if all this stuff was just in a native soil that we basically just created beds on top of a native soil, does it serve any purpose to like till it and bring some of that stuff to the surface before solarizing, to like make beds, till it, and then solarize it to kind of maybe kill some stuff that's in the soil? I don't know if that makes sense at all.
Yeah, I can address a couple of those points. First, I want to start out with the fact that it's affecting so many different crops makes me think there might be a few different soil-borne diseases, because the Fusarium, right, it really picks its lane and will only kill one type of crop. And so you definitely might have had it like on strawberries, for example, but then there might have been maybe a Phytophthora in an area of your field that doesn't drain as well, or something like that. And some of those will affect many different types of crops, and it can kind of obscure, what's happening on any specific crop. And it makes management recommendations really difficult. So definitely the first things I'm going to think about are just what can we do that's good culturally to start with, because things like waterlogged soils or compacted soils will hurt you no matter what the soil-borne disease is. And so in terms of tilling or something like that, I would definitely make sure that you have good, or rather not a lot of soil compaction, right? And so if you need to till on your scale in order to break up the soil, that can be an option. If you can broad fork, that's another option. The transition from till to no till can sometimes lead to a little bit more compaction before a grower has all the systems dialed in. And so if you are struggling with diseases, I would definitely keep an eye on that. In terms of planting cover crop, I really like cover crops. It's really hard to leave a sea soil be bare all year. For Fusarium in particular, leaving a soil bare will help lower the populations faster than most other treatments. But on the flip side, from what little data we have, it seems like growing anything that isn't the primary host for the most part of that Fusarium will help lower the levels of pathogen in your soil. So if you have Fusarium of tomato and you plant, a rye cover crop or something like that, you still are gonna, you haven't accidentally raised your levels of the pathogen, and it'll probably still help. And if you're getting a lot of other benefits like nutrient cycling or reduced compaction from that cover crop, it probably outweighs the any negative benefits that would arise, but it's so hard to generalize on that kind of thing. Let's see. In terms of raising your beds, that can definitely be a great way to reduce compaction. So if you have the ability to do that, I know around here raised beds are the standard vegetable production technique, and they don't use permanent beds. They'll flatten it and re-raise them each year. But doing that, you can get a few extra inches of really well prepared soil, and giving them even a good head start can help them get to a productive place before you start getting disease symptoms. It's really hard to eliminate disease in 1 year. And so, definitely be keeping this in mind, and which piece of the disease triangle can you be combating with each of your management decisions. But the easiest place to start is always the genetics. So if you have something that was giving you a lot of problems, or that you have confirmed the specific pathogen that was getting it, like I always come back to this because it's what I work on, but if you had Fusarium in your strawberries, get a Fusarium resistant variety, and you've eliminated that consideration from your other management tactics. Like, you're good to go Fusarium wise. Because I think about this all the time when I'm planting my own garden, that's often where I start from, because I feel like otherwise I'd be a bad plant pathologist without that. And there are so many other considerations when choosing seed. But again, if you're having specific crops that you're getting hit on with a specific disease, that's the best place to start. And some of them have pretty good disease packages. Is there any other specifics I can get after on your question?
I guess just the mustard as a cover crop. Yeah, if there are like biofumigant properties to that.
There definitely are. That is something that has been shown to reduce diseases and nematodes. It's really hard to grow enough of it. And so if you do want to use that for a disease fighting purpose, it's really important to grow that as well as you can. And I'm not going to pretend it's any kind of miracle. It's something that can knock back pathogens a little bit. And again, it really depends on the pathogen. It can go a little bit either way with Fusarium, because it will eat just the organic material sometimes, especially if you don't have enough of the material to really generate the toxic gases. Some people have combined that with tarping to really hold the gases in. And that could increase your efficacy on that. But again, it's tough to deal with Fusarium, in particular with the organic amendment options. There is a company called Farm Fuel that I know does this commercially for Fusarium, or for disease suppression in general. They might have more information for you in particular. I've never worked with them and can't recommend them. But I do know that they exist and might have additional information.
Thank you.
I had a quick question building on that about if he goes and tills, would he be spreading Fusarium from one area to another? And I did actually look back up. So John, I had Fusarium and Pythium in my notes for damping off from the lab results that you got, but there might be other things too. So to Gabriel's point, there's probably a lot of stuff going on. But yeah, so would tilling, like, love the idea of improving aeration, making a really nice seed bed, raising up the soil a little bit. Would that process though, like, move it around more?
So one thing I want to point out here is if you see on field one on the bottom left side, a lot of this fusarium damage is sort of in lines, and
Whoa.
it's sort of, yeah, it's sort of indicative that you can, and again, this is just observational from my point. I did not conduct this experiment, but there are instances where you can drag pathogens into your field. And so it's something to consider. If you know that you have a lot of pathogen in a spot and you move through it, right, you are pulling that pathogen with you. And so it's really important to stack our disease fighting tactics. And because, the top of this field and the bottom of this field were treated the same, and you can see some of the streaking in both halves of the field. But, by having good conditions with like cooler conditions where the fusarium wasn't favored, it wasn't as bad. And so if I walked into field one, I would say both sides are affected by fusarium. So again, it's really hard to eliminate it. But we want to avoid the situation where we're really pulling the disease. That being said, it's not in all contexts. On field three, you can see it's kind of blotchy, and that might be a disease pocket that just grows over time. So it's not something that we can say for sure. But looking at field one, I would definitely say it happens sometimes.
Thanks. That is just a great like illustration of that. And interesting that it's not necessarily consistent though, and that patchiness in field 3 is interesting. Thank you.
Yeah, absolutely.
I realize it's 3:15. I still have time. I don't know if you guys want to stick around for another 10 minutes or so. I think this has been really productive. I've learned a lot, but yeah, I'm happy to just hang out for whoever, wants to stick around.
Yeah, I have a couple more minutes for sure.
Thanks.
Are we gonna oh sorry.
Oh, go ahead. Go ahead.
The well, I know this is Fusarium based, but would the same principles apply to something like Phytophthora? Because that was also something we were kind of like on the fence about. And Ellie, I sent you those other lab results, I think, from a different test that had shown some Phytophthora in the field. So I mean, are we kind of thinking in the same way about those kinds of things?
Many of the things apply to multiple pathogens. I would definitely start, kind of at the top of the list for what you can do about multiple pathogens at once. For example, while they may not have bred every crop for every disease, it's like if you can find a plant that says Phytophthora resistant or something like that, it's like that's always a good place to start. Granted, you'd have to find something that has each of the diseases that you want to combat. And there is some kind of strategy there for which disease packages you'd place in each part of your farm. And that's where you start looking at what favors each disease on that triangle. For example, Phytophthora, is called a water mold and it loves free moisture, just absolutely loves it. So if you have any low-lying areas in your farm that are getting some sort of soil disease, that might be my first guess, but it's really hard to tell just visually. But so you might want to be targeting, it's like, all right, I'm going to plant my Fusarium resistant plants kind of higher up where the soil temperatures will be higher. And then where I'm getting a lot of water logging, that's where I want to focus my Phytophthora management tactics. That being said, it's like the overarching disease or the cultural practice of like really good drainage, no water logging, or plenty of oxygenation will help with both diseases, just maybe in a different way. Some of the more specific things like what pH do you want your soil definitely can change. For Fusarium, you generally want it a little bit higher, and that's the case with some other diseases too. But and I can't come up with a counterexample off the top of my head, but there are other diseases that get worse at higher pHs, or maybe you'll accidentally tie up too many of your nutrients, and so that's something to keep in mind too. And so targeting, what's the ideal for your crop? So maybe something, above 6.2, but under 7. Often maybe closer to 7 if you're trying to combat Fusarium can be good. And that comes in with proper liming, right? And that lime will add calcium, which has been shown to work against Fusarium. It's also been shown to work against Phytophthora, so that's a good place to start. But something kind of specific about like the ammonium I was talking about, not all diseases show that. Some of them are the complete opposite. And so you really have to get your diagnosis. And Fusarium loves ammonium, verticillium loves nitrate, right? So where, like, what are we dealing with here? I don't know what the case is for Phytophthora. But the overarching principle, over-fertilization or under-fertilization will exacerbate all the diseases. So, you know, can we get soil tests, make sure we're maintaining nitrogen levels at a high enough level for crop growth, but not too high? They sell some quick test strips, and UCANR has good guidelines for different crops for like kind of in-season nitrogen testing that you can do yourself. They're just like pH strips where you do like a little dip and read the color. So something like that can help. It adds a lot of complexity to your management, especially in a farm like it sounds like you have, where you have many different crops. But it might be a place to start, just to get in your mind, am I way under fertilizing? Am I way over fertilizing? Do I need to like do a little bump here? So yeah, starting with those basic principles will be the best way to get a lot of diseases at the same time. I'm really a fan of proper nutrient and irrigation management for just general soil-borne disease control. And you may have been able to pick up on that by the fact that I've mentioned it 100 times already. And there's a software called Crop Manage through UCANR again, and that one's for also irrigation. And so you can kind of measure how much you've been applying and kind of compare it to maybe what the software recommends that you apply. And that can give you a good baseline. Even if this isn't something you can do every day, it can kind of be like a key in, like, should I be running my irrigation for closer to 2 hours or closer to 1 hour or closer to 3 hours? I understand too that adding all these things in adds so much complexity to management. And I'm not going to just sit here and pretend you can check these things every day, but just keeping it in your mind of like, what part of the disease triangle can I combat, can help you get at multiple pathogens at the same time. I hope that's helpful.
Yeah, for sure. Does anyone else have a question? Because I got another one, but if you guys want to jump in, please do. No. So like with this area that we have cover cropped, is there like a certain, I mean, you know, in an ideal situation, you're able to move the whole operation to another place, but you know, it's like you can only do that so much unless you just have endless land, right? I mean, we might be able to pull it off for a season, but then we're gonna probably end up going back to this other piece at some point. So is there some sort of window we're looking for in terms of like in terms of like how long we leave that fallow or cover cropped, and also like what is the re-entry strategy for, like, you know, testing to kind of get it? Do we just like do a strip where we knew there was some disease and try to plant some susceptible plants and kind of see, like, I mean, how much of like, is that a year? Is that 2 years out? Is that kind of like, you know, what's kind of like the ideal? I guess as long as possible would be the ideal, but you know, I mean, it's like do I wait like do I leave it fallow for a year and then try to start replanting and kind of like testing the waters, or yeah, I don't know.
Yeah, there's definitely a couple ways that you can approach it. And again, it's based on your context. But the good news for Fusarium in particular is that it really does look like the amount of pathogen in the soil starts going down quickly. So even after 1 year, there is much less Fusarium in the soil, and you might see some benefits after that. The general rule of thumb is we would really like to see 3 years without a host crop before you come in. So, you know, a 3-year non-strawberry growing rotation, for example. And that's going to give you pretty good results. depending on your system, right, breaking ground and like making new beds can be a really labor intensive thing. And so you could try like switching back and forth between your locations every 3 years, for example, and that'll give you a little bit more time to rest one area while you're, still, you can use the same irrigation infrastructure, for example, for that 3 years. So you're saving a little bit of money and time there. That's a general guideline. And obviously the economics of the business have to come first. In terms of like testing for Fusarium in the soil, you can definitely do that in a lab. Not all the diagnostic labs will test for all the pathogens. In order to do this, these are molecular assays. So just like they were doing at the beginning of the COVID pandemic, where they'd send in the samples and you'd have to like literally have a technician, move every single sample to a different tubes, and it's a whole process. So it can be kind of expensive. You definitely can do your own assay in a greenhouse, where Ellie was saying never use field soil for a transplant. But if you want to see if you can kill it, that's one time where you can. So you could purposefully get really susceptible cultivars, plant them in like a little transplant tray. And if it just gets absolutely destroyed, that's probably a good indication not to use that. And you can even take that plant once it's died and send that into a diagnostic lab, and they can double check what the pathogen is that killed it. But ultimately, a 3-year rotation generally is really good for Fusarium.
Okay, awesome. Thank you.
Mm-hmm.
I love that idea for testing it out after like 3 years or however long. Like, does it kill the plants? That it used to. It's a really cool idea. And maybe throwing in some like resistant varieties too, to see like, do they stand up to it? And then you have plant material you can send off to your lab if you need to. Love it.
Yeah, that's a great idea, throwing that resistant in too. If you have one that dies and one that doesn't, one's susceptible and one's resistant, you might not need a lab. It's like, all right, I've answered my own question there.
Any other questions or thoughts on any of this stuff? I know, like crop types wise, we're talking about strawberries, brassicas. Are there other crop types? Have people seen this in tomatoes? I saw it at one farm. The farmer's not here, but okay, so you have like how prevalent is it? Like every year, same place? Is it growing, across the field? What does it look like?
That's a good question. I mean, we grow many varieties of tomatoes. We normally have like 2 successions that are in big blocks. Certain varieties are hit harder, and we need to probably pay better attention to like, where it's creeping in and stuff. It seems like by the end of each succession, like after they've been in production for a long time, they're really struggling with this. Yeah, I'll need to kind of take better notes this season and report back, but it seems like very consistently tomatoes for us, it's an issue.
Yeah, that's interesting. There's another grower who's not here, and it looked to me like some of the varieties he grew a ton of different varieties, and it seems like some of them suffered a lot more than others. So yeah, that would be interesting to get a better handle on like which tomato varieties are actually grown around here, and then which ones get hit really hard, and which ones are maybe okay.
Yeah, we're also pretty like stuck in our ways in terms of varieties. And this year, since we're trying to grow our first succession in hoops for the first time, we're trying different varieties that have, different disease packages. But I think it might be good to, branch out and try different varieties with different resistance. Would be interesting.
Awesome.
Taking those notes is definitely a great way into the Arn Farm research. And to a certain extent, as a scientist, I have to be able to report this is the exact pathogen, that's hurting this exact cultivar. But if you're planting out a bunch of tomatoes just, to sell them, at least taking the notes on which cultivars are going down first can let you like drop them from your rotation. And then maybe your level of disease just kind of naturally goes away as you get rid of the ones that aren't performing. And then you don't necessarily need to know which disease in particular is getting you, even though I would still recommend that. But just over time, you're kind of evolving your crop selection toward the things you're having problems with.
Yeah, and like you said earlier, if anyone ever has questions about like setting up a field trial and like what does randomization or replication mean, or like, you know, you feel free to reach out anytime. This is like what we do. So yeah, and it, like you said, it could be simple as long as you just have a control and a treat, you know what I mean? That doesn't have to be super complex. So it's always nice just for comparison to see like what really works and what doesn't. But yeah. Any other questions? Rachel, you look like you're gonna say something. Maybe, maybe not. Oh no, you're
You're
muted. I think you're muted.
I was muted. I just, yeah, I'm kind of just wondering whether I should test, because I had so many different crops that were affected by something. I'm wondering, like, the strawberries, I think just it came up, it was just Fusarium on the strawberries, but whether I should get other crops. I mean, at least I must have multiple kinds of Fusarium, but it could also be multiple different fungal diseases. So I'm kind of just realizing that I don't know if there's other diseases in the soil, and kind of how to, like, that's a little bit of a challenge. And I mean, the soil solarization seems like it's good for everything, and adding vermicompost is good for everything. But like how big of a problem it is that I don't know if I have other diseases. But
You know, I like to recommend testing versus guessing. It's if you're going to ask me, should I test or should I not test? I'm always going to tell you to test. It's up to you to determine kind of the economics of that. The samples on an individual basis aren't that expensive, but if you're going to send in a dozen samples, you know, it does add up. So yeah, do with that what you will. The soil solarization is very broad spectrum. The vermicompost seems to be very broad spectrum, but there's not a lot of disease. It's not a magic bullet, but I think it is a reasonable follow-up to something like the soil solarization, just to help add in some microbes, which you know are not going to be pathogens. So
Yeah, and I would just agree with all that and just start with, you know, all of the things that you know will benefit and address any soil-borne pathogen. Like, you know, I know you're on a small scale. If you can broad fork, that's great, or find some way to improve drainage. And I also know it sounded like it flooded at your site previously, so I'm thinking Phytophthora would have loved that. So it is definitely possible you've got some other stuff there as well. And yeah, I think testing, if you can, if it pencils out, is great. But I also really wanted to mention to everyone that through UCNR, you can send samples to certain specialists. So like we have a specialist for tomato pathogens. Her name's Cassandra at UC Davis. And you can, if you go through me, you can send it to her and she'll analyze all of it for free. And so maybe that's like 3-4 samples or something. And you don't actually have to pay for that, which is great. And you get to work with me and I can help you interpret it and think through next steps. So that's cool. We have that for tomatoes. We also have a Keith Escalon for strawberries. I don't know if we have anyone for brassicas. I haven't been able to figure that out yet. I don't think so, but those are two resources that are pretty great. And like Keith is who analyzed your samples, Rachel. So he's fantastic. But the thing is, it's free, but it's slow because it's not a commercial lab. It takes a while. And sometimes I have to check in and remind them. So less fast, more free. But you can also look, you know, ALNL Labs is great. TriCal Diagnostics is great, and they're going to give you more specifics. So TriCal will say, you know, genus, species, and maybe even more details beyond that. It's more expensive than ALNL in my experience, but you know, any testing is going to be helpful. It's better than, I would just agree with Gabriel. It's like better than not testing. But if you want to wait, I would say you could just start with doing all the practices you know will help, and then see what happens. And if you start seeing those symptoms again, that's when you have fresh plant tissue that has disease symptoms. And you're like, okay, there it is. And then you can send it off to a lab if you so choose. And that might be 1 sample, that might be 5. It just depends on like cost and like, you know, is it worth it or not?
I don't think I really have any disease. Like it does seem like whatever it is goes away in the winter.
Are you growing anything right now, or?
Well, I just I mean, I cover crop, but there's just like overwintering brassicas and fava beans, and like I don't really see.
All right.
Yeah.
Okay.
Yeah.
Well, good. Yeah, I mean, that's great. You're keeping an eye on it. And yeah, if you do start, and you also know what to look for. So if you start to see it,
Or
Yeah.
nothing, it's, if there is anything, it's much, much more minor than what I saw in the like spring through fall.
Okay, yeah,
One
that's
thing-
interesting. That gives us clues about temperature maybe, but sorry, go ahead, Gabriel.
Oh, no. It's another nice thing if you've planted all of your cover crops like in a uniform mix, you know, you can kind of, it almost acts like a bar chart by itself, right? Where you have some that are really tall and some that are much shorter. And so that can clue you into those problem areas. And maybe don't plant your super high value crops in the areas that are suffering, that the cover crops are suffering in, because, you know, you're setting yourself up for some failure there.
So Rachel, were you done?
Yeah, I actually have to go. This has been very helpful, but I have to go. I have to go, even though I want to keep listening. But thank you. Yeah.
Yeah,
Yeah,
no
thanks for
worries.
coming.
This will be recorded and I can send it out and send out the PowerPoint. So yeah, no worries. Thank you so much for being here. I appreciate it. Bye.
Yeah. I just got one last question about Ellie, this might have been in the material you gave me, but with the solarization, Gabriel, just if you have any thoughts about light transmission for the plastic, like because I did try solarizing for weeds. I used a 4 mil plastic. It became super brittle and like broke into like a ton of different pieces over the course of the season. So I was thinking about jumping up to like a 6 mil. But then there's a question of, you know, what the opacity of the material, like to get the most effective heating. Is there like a certain percentage that we're looking for, or do you want it to be like completely clear for the kind of like the max effect?
Generally, more transmission is better. It really gets a better solarization effect just by letting the light in. But that's honestly a question for like a plastic sheet manufacturer. They might have something that's both thinner and not going to become brittle for you, because some of that's in the composition of the plastic itself versus the thickness. And so I'm hoping that you don't have to spend the money on a 6 mil plastic. But yeah, I might poke around on some of the agricultural supplier websites, and maybe they'll have more information than I have off the top of my head.
Okay, cool.
Yeah, and I can look into that some more too, and see what I can find.
I was gonna reach out to Jim Muck, who does the garden at UC Davis. I guess they've done solarization up there, so somebody told me that. But he's a great guy, and it seems like they might have some experience with like broad scale solarization practices.
Yeah, it seems like a great idea. If you can get someone who's done it before successfully, it's like they might just have all the hot tips, because I've never personally solarized my garden, right? So it's hard for me to point out all the tricky pieces that are going to get you.
For sure.
Yeah, and I like the idea of finding material you could reuse, you know, and that won't break down to a million pieces in your soil, ideally. Yeah. So there's got to be something out there. Yeah.
Sure.
So let me know what you find out and I can look into it too. But
Absolutely.
okay, well, we should probably wrap it up unless there's any other thoughts or questions. Nope. Okay. Thank you all so much for being here. This is awesome. I've learned a ton, and thank you, Gabriel, for your expertise. And thank you, John and Susanna, for being here. And it's nice to meet you, Susanna. I don't think I've met just only over email, but
Yeah, nice to meet you. Thank you guys so much. This has been super interesting.
Yeah,
thank
thanks
you
for
guys.
having me.
All right.
Yeah. Okay, well, have a good evening, everyone, and don't hesitate to reach out. Actually, should we put our emails in the chat real quick? Actually, Gabriel, would you do that just so that they have it? I know you put it in your presentation, but it might be nice to do that too real quick. Thank you.
Yeah, absolutely. I'm always happy to chat. My expertise is definitely closer to Fusarium, but I'll do my best.
Thanks. And yeah, and if you want to do trials at any point, Gabriel, it would be awesome to have you do trials up here. It's not too far. You're from here. It's like, I don't know, 3 hours from Salinas, hopefully not too much of a hassle, but please feel free to come up and reach out anytime if you want like a field trial or something like that. I can
Let's do it.
share it with everyone who attended today. So yeah. Okay, awesome. We'll let you go. Thanks,
Thanks,
everyone. Have a good evening.
Thank you.
Yeah, see you guys.
Bye, everyone.
Bye.